Okay, let's now go through the microbial typing techniques that are enhancing our insight into microbial population, and they are widely used in diagnostics and pathogens related. Marian Freitzleber from Research Group Data Science for Life Science will share insight into some pathogen genomic structure and relatedness using core-genome multilocus sequencing typing. Let's have a look how we can have it in all-in-one platform for the molecular bacterial typing. Good luck, Marian.
Hello and welcome to the presentation of our Shiny app, PhyloTrace. PhyloTrace is not a classical unidirectional bioinformatics pipeline. It's rather a platform to track, manage, and analyze bacterial strains on a genomic level. Here we focus on a specific kind of typing method called core-genome multilocus sequence typing. That's basically a gene variant comparison of a subset of commonly expressed genes of a specific bacterial strain. This method can give insight on genomic relationship, outbreak dynamics, and detect new gene variants. PhyloTrace can be launched from desktop like that. So when the app opens, we have basically two choices. Either we load a pre-existing local database containing the schemes we have worked with before, or we create a new one, choose a custom path, and select it. Building a new database from scratch means that our browser is empty. We first need to download a typing scheme from a database. PhyloTrace currently is connected to the cgMLST.org publicly accessible database, comprising 40 of very relevant clinical bacterial strains. Here I loaded the Bordetella pertussis scheme, which contains all the gene variants of this bacterial strain. Now to type a first assembly, we need to go to the tab Typing, and here we select an assembly file. Now we need to declare all the basic metadata which is connected to this assembly file. These variables will later, after the typing, appear in the database and can be edited in retrospect. Once I click Start, I basically launch a very fast alignment algorithm written in C, which scans the assembly for the genes present and checks which gene variants can be found in this bacterial sample. Alternatively, we can switch to the multi-typing mode, which is allowing us to select a whole folder of assembly files, which are all typed automatically without that we have to start them one by one. In this case, we have already started a single typing process. Therefore, it's not allowing us to start a multi-process in parallel. I fast-forwarded the process to the end. The whole typing, in this case, took about 5 minutes. Before we can view the new database entry, it is indicated that there are missing values, which is common because sometimes not all genes can be recognized. This tab allows us to determine how to deal with these NAs for further analysis. Now we could proceed and add more and more entries to the database until it, for example, looks like this. This overview allows us to browse all the assemblies we have typed over the course of time. There are many options to edit the database. For example, we could change metadata or delete a whole entry. We can save the database with our changes. If we decide otherwise, we have the option to undo the changes and go back to the previous state. We could also compare individual loci or download the table to an external file. The addition of custom variables is also possible. Here I selected the variable to be numeric. Afterwards, I can go to the database and feed our new variable with values. In this case, I prefer not to save the changes. Instead, we head over to the Scheme Info tab, which is showing us information about the current scheme, such as how many genes are included. Here we can search for specific gene products, such as kinases, and for example, sort them by length. This tab shows a distance matrix heat map with all the genetic distances between the assemblies we have in our database. And now we proceed to my favorite part, which is visualizing the genomic relationship of all our entries. The resulting graph will be a tree-like plot, and we can choose between the three algorithms: minimum spanning, neighbor joining, and UPGMA, which will all lead to a tree plot. In the database browser, we can select which entries should be included in the resulting graphs. For example, I could select only the first 16 entries, go back to visualization, and create the tree again. The user has the possibility to change the appearance of the graph. We can customize plot elements in great detail, for example in color, size, or add variables. MST plots, for example, can be dragged around and zoomed in and out to achieve an optimal look. Here you can see some screenshots with examples how MST plots could also look like. I would like to showcase how to produce a sophisticated neighbor joining plot in real time. We have the chance to choose an alternative theme, for example a circular one, and it will automatically adjust to the quantity of isolates contained in this graph. But for now, I will choose the round-rected theme. What I also like is adding a root edge to distinguish the root from the other branches of the tree and make it dashed to distinguish it even more. Now we can also add several elements, such as tip points. Simply by adding tip points, we actually win nothing, but we can map metadata or other variables to this element and eventually reveal hidden patterns that explain the underlying genomic relationship. Here I want to highlight in which city the isolates have been collected and select a color scale which is appropriate. To increase the visibility, I will change the opacity to the value of 1. So let's assume that these isolates here have shown a resistance gene and we want to highlight it in our graph. We can toggle the node view, remember the parent node number 99, select it, and it will display and highlight this parent and all the daughter nodes. Now we can also label it with a custom label. We can type resistant, and now we have to position this custom label to the right place. So this is not quite the position I want. I will increase the horizontal position again, decrease the size a bit. Now I want to show you how to add even more variables. For this, it's better to change the layout a bit and align the labels to the right. Now I will head over to the variables again and add a tile, which is basically a bar. Here I choose a patient age variable. And I select the Cividis color scale to highlight very young and very old patients. I will also adjust the position to the right in order not to superimpose any elements. I also added a second tile carrying a gene expression variable and fast-forwarded it to speed everything a little bit up. The last step left is to position the legend so that it doesn't overlay with other elements, and the same is done with the tree scale too. And there we have it, a publication-ready neighbor joining tree. In addition to the genomic relationship, it carries much more information. In this example, 3 variables, patient age, city of collection, and gene expression values, which allows to grasp the bigger picture. In these other examples for neighbor joining trees, you can see that also heat maps can be added. It can also have a circular layout, as for these trees created with the UPGMA algorithm. Once you are happy with the appearance of the plot, you can save it in different file formats. Moreover, you can have yourself a whole report generated. Here you can checkmark which elements you want to include and add information such as the operator, the institute you are working with, and comments. And that's the last step. I will now download the report. As selected before, we will also have a table here containing all the bacterial samples that are included in the plot, as well as analysis parameter and information about the cgMLST scheme. That's it. Thank you very much for listening, and feel free to visit our GitHub and the website and try it out by yourself. Have a great day.
Thank you so much, Marian. We have one question. Your app looks like a very complex app with many data visualization features. Can you share how you manage such a big app? Do you have any tips, maybe?
Yeah, so actually this is my first serious Shiny app. So I don't know if you asked the right one about managing that. So far, I would rather call it a mess, but we plan to use the Rhino framework and modularize the app in retrospect. So that's the plan, and quite a big one too.
Okay, thank you so much. And thank you so much for sharing your app. If you have any more questions to Marian, please don't hesitate to put there in the chat a comment. Right now, we're moving to the next presentation.